Hello everyone, welcome to session 4. Today we are going to be covering some trace minerals and some ultra trace mineral. Objective is that we understand how they are absorbed, in which form they are absorbed, what are the cofactors or other nutrients that support that pathway. I would also like to uh, kind of emphasize their role in our body. Then talk about some of these nutrient interaction. We still don't know why and how, but we'll try to pinpoint the important ones. Then you will notice that we are discussing iron today. That is part of your assessment. So my aim would be to discuss a couple of those questions here. And then we are having a webinar next Tuesday. So I will be posting the link about that webinar tomorrow. Okay. And just a gentle reminder that we are recording this session. So keeping yourself mute. And after every nutrient, we will take, a couple of minutes break, and then we can discuss our questions. Right. So now these trace minerals, they are called trace because, for the longest time, we didn't know that they are stored in the body. We assumed that they are required in micrograms. And till 1960s, we didn't have the technology to measure their concentration in our blood or serum or urine. So in the starting, they name it trace because they didn't know how to measure them. But now we know that some of them are required uh, up to about 100 milligrams per day, whereas the ultra trace ones, they are required in micrograms. So think nutrients like boron, they are needed in less than 1 microgram amount in our body. So what do they do in our body when they are present in such small amounts? Mostly, you will see that there is a term written here, metalloenzyme. So most of these ions, they are present in as a structural component of an enzyme. And secondly, they can work as a cofactor also. So there are some of these enzymes written here. I would like you people to remember the first one. We will be repeating it a couple of times in another chapter also. So it is called as superoxide dismutase. So some of you would recall superoxide is a free radical. So it is created in our body when we are getting rid of toxins in the liver or our body is fighting any infection. And then superoxide dismutase is our body's endogenous antioxidant, and it is made up of copper, zinc, and some other proteins. So you can see copper and zinc both are structural component of this metalloenzyme. Okay. Then catalase again is our endogenous antioxidant, and it requires iron. And then we'll talk about zinc and manganese in the coming up slides. So that's just an overview of how important they are despite being required in micrograms by our body. Okay, coming to that tricky one, forms. But I'm hoping that you people have become kind of expert now that there are various forms of all these nutrients, including copper. So we have a cuprous and we have a cupric. Cupric is the one that appears in the food. That is the form present in food. And would anybody like to comment on different food sources of copper? Let's see. So we have Jane telling us that it's the liver. Yes, very good. Organ meats, they are very good source. Oysters as well, and nuts and seeds. Okay. So copper is required for healthy nerves, bones, collagen. It's required for our skin pigment, melanin. And mainly in all those functions, it is acting as a cofactor. So I've talked about this first point. Coming to this second point, we will be talking about that when we discuss iron. But, you know, because of this valency, this free electron outside, these minerals, they help each other out. So when we have to get iron in our blood, we need copper, because copper is able to give this extra electrons to iron so that it can move around in our body. So need to remember these two names: copper containing enzymes or transporters, ceruloplasmin and hephaestin are critical for iron utilization in the body. Then when tyrosine is getting converted either to thyroid hormone or neurotransmitter, we need copper dependent enzymes. Okay, so now let's dig in to the absorption. So the same story again, because copper is present mainly in the protein foods, it is bound to all the amino acids. And in the first step, hydrochloric acid and pepsin are imperative because they release the free copper. Now that free copper from the food is in the form of cupric, which is with the valency of 2 plus. Okay. So what does the body do? Body has to convert it to the cuprous one, because that's what body loves or like to absorb. So for that, we have got this enzyme, cupric reductase, and you can see that it's written ferric also. So what does that tell us? So that, yeah, some.
It acts on iron?
Yes, absolutely right. So it's the same enzyme, or and if cupric is coming, it will work with cupric and convert it to cuprous. If ferric is coming to the brush border, it will convert ferric to ferrous. And both of them need an acidic environment. That's why vitamin C is beneficial for their absorption. So let's come straight to the diagram. So you know what's happening here in the first step. Then here you can see that if there are some enhancers present, apart from vitamin C and low pH, even some amino acids, glutathione, they also help because they convert cupric to cuprous. After that conversion, cuprous has a specific transporter, simply named as copper transporter 1, that has a high affinity and it takes copper inside the intestinal cell. However, there is another one which is called as DMT1. So it is dimineral divalent mineral transporter. So that means all those nutrients or minerals that have a particular number of electrons, it can take all of them. It can take zinc, it can take manganese. So that's why, because it has this job to carry so many of them, it has less affinity for copper, and there is a possibility of competitive inhibition as far as DMT is concerned. Now remember, whenever the cell is coming to our amazing intestinal cell, sometimes they are growing, they are fighting some infection, they use all these nutrients. Here also, copper can be part of metallothionein. So that's another storage form of copper, and this is the one that fights infection. It is mainly in the liver, but even intestinal cells have the capability to store copper. And with copper, it also stores zinc. Both copper and zinc, they are together in this metallothionein. So that can happen inside the cell. Then copper can use itself. It can be used by enterocyte to do its own function. And look here, copper, because it is quite oxidative, because of that free electron moving around, it can interact with other things in the intestinal cell and get converted to cupric quite easily. That's why they need a ferry ride. Okay? So they have these chaperones. These are the transporters specific for copper in the intestinal cells. They are also present in the liver also. So when copper is moving about inside the cell, it is always bound to chaperones. And you have the name of some of these amino acids that are doing that role. So it is glutathione or etox. So for now, try to remember only the word chaperones. Then copper comes at the basolateral layer. Here we have got energy dependent transporter called as ATP7A. This one transports it to the blood, and in the blood, copper is always bound to protein, albumin or macroglobulin. Then it goes to the liver where it gets attached to ceruloplasmin, which is its main transporter in the blood. Now, just staying with this last point on the slide. Now, some of you might have heard about Wilson disease. So we are aware that there may be a genetic mutation in the gene that is making ATP7A. So this transporter is specific to the intestinal cell, and in the liver, copper has another one, ATP7B. So whenever there is gene mutation in any of the genes transcribing from either of them, a person may have Menkes disease or Wilson disease. Quite rare, only 1 in about 40,000 people can have it. And it's quite visible when a child is about 2 to 3 months. They may have neurological problem, kinky hairs, vascular problem. Sometimes intravenous copper helps, but we don't know how to prevent or how to address this condition. So it is quite fatal. Okay. So another name to remember is that in the liver, we make ceruloplasmin, a transporter that carries 6 copper atoms and it moves around in the blood freely. So if every Friday I am in a habit of abusing my liver, by consuming alcohol, or I'm working in an environment that is exposed to a lot of heavy metals, such as lead, then there might be deficiency of ceruloplasmin, which can translate into deficiency. Now, agonist, because these amino acids, they act as chaperones, they are considered agonist. They bind to copper, prevents its oxidation, and transport it through different organs in the body. You would now understand why organic acids are agonists, because they are providing a conducive milieu in the gut for cuprous cupric forms to interchange. Okay, now look at the antagonist. I just told you that copper is stored in a form called as metallothionein, and there zinc is present along with copper. So why copper and zinc are antagonists? Has anybody? Yeah.
Is that because they share that transporter? They share
DMT.
this
Yes.
competition, the DMT1?
Yes, Kathleen, that's one of the reasons. Anybody else? Competition, Amanda. Yeah, that's the point. It's tricky. Like, I was trying to understand why this happened. Then I was looking at supplements. Some of them have both of them together. So my biochemical mind was very confused for a little while. So what I understood was that it happens only if somebody is taking a supplemental dosage of, and what is the upper limit for zinc? Sorry, just played with my mouse. So the upper limit for zinc, if you look at the NRV's value, is 40 milligram. So one of the, not one, there are 2 or 3 research studies done in humans which told us that if somebody is taking more than that upper limit, which is 40 milligrams and above, that actually tricks the body, because all that zinc is going to metallothionein and it has to has copper with it, otherwise it cannot do its function. So in a way, body gets into this mode that there is too much zinc because we need metallothionein to fight the infection, and that's why we need to bind copper with it. So it's happening even at that level. Does that make sense? Like, so that's the second reason why it is happening. So now I want you to kind of brainstorm, how would you address a client whom you want to give a zinc supplement?
Nancy, would you give a copper as well? I'm not sure, I'm only guessing, but would you? No, you wouldn't give a copper?
Yeah, let's discuss it. Like, let's wait for others to, you know, say anything. We are learning, guys. It's okay. Yeah, so Kathleen is of the point of view, she may want to give copper. Anybody else? So we are assuming, number one, that this person is deficient in zinc as well as copper. For that, we need to have some signs, we need to have some blood test and things like that. Second is that copper can be toxic. High amount of copper can be toxic. So have to be really mindful to give divided dosage. That's what my understanding is. So maybe give only 30 milligrams of zinc per day. Don't go to that upper limit. And then add on some dietary sources as well. Yes, Michelle, that would also make a good point to add vitamin C, because that will ensure that most of the zinc or copper that is going is getting absorbed. So working with smaller dosage, unless there is a critical phase where you want to give a high dosage, but don't go above 40 milligrams. So, and be mindful, we might not need copper unless that person is deficient. Okay. Oh yes, Amanda, definitely. Good amount of proteins would help. Yes, high biological value. Yeah. And you know what happens? Sometimes people come to us with, okay, now I'm discussing, sorry, a different point, iron. Here it's written large amount of iron can also affect absorption. So that's again, there is a competitive inhibition, but one of the functions which we discussed a couple of slides ago was that copper is required for iron absorption. So there is a kind of a contradictory statement again here in these minerals. And you know, sometimes it does happen that there is a client, I see that they have low hemoglobin, iron deficiency, continue to give them iron supplement, and it is not working. And then we realize it might be copper deficiency, which can be visible by simply same symptoms, microcytic anemia. And actually it is copper deficiency. Okay. Quite tricky, but just being mindful that in small amounts, in physiological amounts, these minerals, they help each other out. But if there is too much, they may actually phase the other one out completely. Okay, comments and questions.
Nancy, can I just ask you, was there, I don't know if they're still made, but the pipes were made of copper, weren't they, that we sort of used to get our water and things through?
Yes, definitely. Yes, there is a chance. And you know, like in my mother would cook in copper utensils. And so little, and in fact, we now have all the glasses and servingware coming in copper. Now, I think that's happening because we have depleted all the copper. So that little bit of copper is important. But yes, it can come from the source in which it is being moved around. Yeah. Did I answer you or did I digress?
No, you answered me. Thank you. I just, Nancy, was there times though when people did have quite like copper toxicity at all from that, or they cooked with the utensils and the water was coming from the copper pipes? But you've sort of toxicity from.
Can't recall, but again, the times that we are talking about, I think humans were not even aware about
copper, yeah.
They were not aware about what toxicity is happening. And lately, past 20, 30 years, we have more choice.
Yeah.
Things have changed. So
yeah. No, thank you, Nancy.
Has anybody heard? Has anything else to share, or do you have any questions at this stage? Okay. Charlie is asking, is the only way to tell if someone's copper deficient through the iron levels, or there are other symptoms or signs you can look out for? There are other symptoms, like we can look at ceruloplasmin level in the blood. That will tell us. And if it is quite a, you know, a tricky situation, then people can also check about the levels of superoxide dismutase, that antioxidant in our body. We can also check their concentration in the blood and in the liver. So that can also give us some indication. Signs and symptoms, can't recall at the moment. Sorry about that. Can't recall that. Has anybody else heard about signs of copper deficiency? So trying to look at from here. Again, fatigue could be one reason. Okay. Frequent infections like zinc, it will be similar, something to like zinc. Okay. So those kind of issues.
Nancy, so the cupric form is the Cu2 plus that we can't absorb, is that right? But the cuprous is the Cu, is that the 1 plus?
Yes, that's the one. Correct.
And that's the one that the body can absorb in.
Yes.
Yes, thank you, ma'am.
So now, usually I've noticed students get confused in the role of chaperones and copper transporter. So just saying that again, that at the brush border, we have copper transporter 1 and DMT1 that are transporting copper inside the intestinal cells. And chaperones are just a co-transporter that help copper move around inside the cell, intestinal cell or liver cells. Okay. So that's sometimes confusion I've noticed students have. Okay, let's move on to iron. You can already see I've added few things, and so iron again, we are concerned about two forms. One is ferric and the other one is ferrous. So ferric is when we have 3 plus as the valency, and ferrous is when we have 2 plus. So what are the food forms of iron, guys? Just a revision for you all. What are the two forms of iron present in our foods?
Nancy, it's heme and non-heme, isn't it?
Wonderful. Thank you, Kathleen. Yes, Megan, you are also right. It's heme and non-heme. So heme is the one which is present in organ meats, poultry, and what else? Any good sources? Egg. And non-heme is the one that is present in plant sources. Any good one? Food sources, favorite ones?
Green leafy veg.
Yes, that's right. And Amanda is also right. Spinach, nuts and seeds. Okay. Think dark green. Remember, I said the same thing about magnesium also. Think when you are thinking iron, think dark green leafy vegetables such as mint. Make a mint dip, coriander, basil. Okay, think dark green. Tracy, you said something like red lentils. So in the break, I'll go, whenever we take our break, I'll share something with you all. It's the black lentils, okay, and black chickpeas that have got more iron than red lentils. So wait for a while, I'll show you during the break. Okay, so now let's move on. So ferric is the form that is present in the plant foods. So remember that one. Okay. And ferrous is the form that is present in heme food. Sorry, non-vegetarian food. So that's ferrous, and it's present as a heme. Heme is kind of a complete protein that has iron in this form. Remember that it's relevant for absorption pathway. Now coming to the functions. So we all know if somebody is feeling fatigue, if somebody is having breathlessness, there is a fair bit of a chance that they have iron deficiency, because hemoglobin that carries oxygen in our blood needs heme. Okay, because it's that heme 2+ that binds to oxygen. So that's one of the major functions of iron. Then in the electron transport chain, again, you would think that means ATP is not producing, so somebody is feeling fatigued. But you would notice that in your assessment question 1, we want you to discuss the role of iron in 2 of the functions in our body, 2 of the important functions in our body. So there we want to understand what is the role of iron, which enzyme it is supporting. So in the electron transport chain, iron is supporting cytochromes. These are the enzymes. Plus, iron is present as iron sulfur complex. And that's how it helps these enzymes. Then various amino acid metabolism such as tyrosine or tryptophan, we are using these enzymes called as monooxygenases. And these enzymes need iron as cofactor. Okay. If you refer to your textbook, Gropper and Smith, it has all these functions listed in its entirety. Then look at thyroid hormone synthesis. Again, if a client is coming to me with hypothyroid, I'll ensure that they are not iron deficient, but it happens together. Mostly you will notice that mostly women, they are deficient in iron, and iron is helping the enzyme peroxidases, which helps the addition of iodine to tyrosine when we are making T3, T4. That's how important iron is. Okay. Right, and again, too much iron could be an issue. And we will again see how important it is for our body to not have high iron. We have methods in place to keep a check, because this iron can be troublesome. And have we heard about hemochromatosis?
Yeah.
Yeah, okay. So that's good. Do you want to comment on it?
I actually can't remember much about it.
That's fine. That's fine. Okay. Okay.
Nancy, I know it's to do with the hormone, isn't it? Hepcidin. You have, I think, with hemochromatosis, you don't have the ability. The liver produces that, doesn't it? It doesn't have the ability to tell the enterocyte to stop absorbing iron.
Oh yeah, yeah. Something. Yes.
Yeah.
So, so I have few answers in the chat box also. Thank you. So Tracy is saying it's iron overload, and Jane is saying it's a genetic condition. Yes, all that is correct. So there is a gene called as I think it's HFT, in which there is no check on iron coming in the blood. So that's why we are storing that iron in the liver and other organs. So there is a heavy attack from iron, and you can see iron can be a pro-oxidant, so it damages the cells. So what do? We have to give a diet that is deficient of iron. We don't have to give enhancers of iron. We have to give actually inhibitors of iron. That is your phytates and tannins and caffeine. So it can be managed. And the point that Kathleen raised about hepcidin, we are coming to that, okay? Slowly we are coming to that one. Good one. Okay. So okay, I've added few things, because for iron absorption, we are going to talk about in 2 steps. first we are going to remember and learn how heme iron is digested or absorbed. So first thing first, this we are repeating actually in all the nutrients. Body utilizes proteases to remove protein component from heme sources. Then second, now in the heme sources, we are getting iron as ferrous, or sometimes the entire protein is coming together. So we have specialized carrier, heme carrier protein. So heme simply gets attached to the heme carrier proteins at the brush border and goes inside the enterocytes. So that's pretty easy. When it goes inside, then body can release iron 2 plus from this heme molecule, and we have got these enzymes there inside the cell. Okay. Now this ferrous is actually quite soluble even in the alkaline environment. So it is not dependent on vitamin C or the pH milieu of our gut. Heme we can easily absorb. So that is the part of the diagram, and the other one I have hidden. So this is what's happening here. Okay. So we have this special protein, heme carrier protein, that carries heme. Now let's come to the non-heme part. Non-heme, again, if I'm consuming almonds, if I'm consuming other seeds such as quinoa, then there are some proteins attached to iron. So we need hydrochloric acid again, protease again to separate iron. Now remember, from plant sources, we are getting non-heme form, which is iron 3 plus. So body needs to convert it to iron 2 plus, ferrous. Again, I'm sorry, I don't know why it's happening, but that's our body for us. That's why we need vitamin C. That's why we need kind of an acidic pH. That helps this conversion. Plus, we also have that reductase enzyme that we talked earlier in regard to copper. That enzyme can also do this work. It can convert ferric to ferrous. However, this enzyme does need acidic medium. Okay. Now this ferrous is absorbed by DMT. So again, that divalent mineral transporter. The same one that copper uses, same one zinc uses, and manganese uses. Okay. So moving on now, both heme and non-heme iron are inside the enterocytes. So heme will release iron 2 plus by this enzyme heme oxygenase. And the other one, and it will kind of form an iron pool. Okay? Whichever source iron is coming from, there will be a pool of it. Intestinal cell can also use it to store it as ferritin, or it can use it to do its own work. If it is growing, intestinal cell is fighting any infection, it will use it. Rest of it will bind to a transporter at basolateral layer, and the name of that transporter is ferroportin. Okay. So remember that one. If you have your tables, you're filling it. Transporter for iron at basolateral layer is called as ferroportin. I kind of remember it that it's kind of a portal for iron. Now it's get a little tricky here. What's happening is that iron now was 2 plus. But when body has to use it, when body has to move it around in the blood, it actually needs the ferric form. Very tricky, but again, how body is doing it. So what do we know now? So it needs one more positive here. So it needs a electron here. We use copper here. So there are copper dependent enzyme called as hephaestin. This enzyme donates that extra electron to iron. So now we have iron 3 plus. And both these steps, they are acting kind of simultaneously. That copper is giving away its electron, converting it to ferric, and then that ferric attaches to ferroportin and comes out. And here it attaches to transferrin and travels to liver and other organs. Questions, or you want a minute to go through these steps, that's fine.
Nancy, what's the apotransferrin? Then I can see what step is that. So it goes through the ferroportin, copper adds the extra electron, and then there's an arrow up to transferrin, but there's also this apotransferrin. What's that?
So that's another ion transporter. And if we have leftover, transferrin has gone about, delivered iron to some places, and now it is kind of without iron. So that also comes in play, get attached to iron, and together they form transferrin. So it's just showing that transferrin can exist without iron. After delivering, it comes without iron, attaches to iron, and form transferrin.
Okay, so sorry, Nancy, is the apotransferrin the form of the protein before it's attached to the iron? That is?
Yes.
And once the iron attaches, it becomes transferrin.
Yes, correct.
Lovely. Thank you.
Good. Anybody else? Okay, good. So, okay. So now I just mentioned that iron can be used inside the cell because iron is very oxidative and it can actually produce free radicals. Okay? So it is always bound by a protein, and Fenton reaction is a chemical reaction where iron can produce free radicals, and we'll discuss it in session on oxidation. So that's why in the enterocytes, it is bound to some protein or the other. And when it is stored as ferritin, it has to be released to the blood. There also we need retinoic acid, and we also need ceruloplasmin there. Okay? Because again, iron has to change form.
Nancy, can I just ask you then? So with this iron, so the heme ion is the Fe2 plus.
Yes. Yes.
It's easily absorbed, but when that goes through into the blood, the body, or before it goes into the blood or on its way out at the basolateral layer, it has to convert it to the Fe3 plus.
Yes.
To travel in the blood. And then when it gets to cells, what form is it? Does it go back to Fe2 or does it stay as Fe3?
Fe3. Actually, body uses both forms. Okay.
Okay.
Yes.
Okay.
So yes.
Yeah. And then plant food is just Fe3 plus, but the body's got to change it to Fe2 to absorb and then convert it back to Fe.
Yes.
Seems a roundabout way, doesn't it?
Yes, that's the summary. Thank you.
Yes. You just think, why not just convert Fe2 to Fe3 and just freely absorb Fe3 plus?
Yeah. Why not? Yeah, that's what I thought. But and again, in the body, you know, in the cytochromes and others, it can use both forms.
Both forms,
Because
okay.
it simply needs other minerals such as copper and zinc to take that extra electron to change form, or vitamin C. So it can work by using both forms.
Thank you, Mance.
So you can make, like when it is making hemoglobin, it will use iron 2 plus. Okay, because in heme, structurally, it has iron 2 plus. But some other places, it can be iron 3 plus also. Okay, so now we have iron moving around in the blood with transferrin. Okay. Coming to another important point on which your question 3 of the assessment is based on. We now know that iron is a pro-oxidant. It can harm the body cells. Therefore, at the level of ferroportin, body has a mechanism in place. If you know how body does it, body measures or senses the transferrin saturation levels. So if the liver gets the message that there is too much iron, there is too much transferrin in the blood, it stops the production, it increases the production of another protein called as hepcidin. So it is different from hephaestin. This is called as hepcidin. And what hepcidin does is it goes to our gut, it goes to the brush border, and it kills away. That's the layman word of saying it. It degenerates ferroportin. That means no ferroportin, no place for iron to come out of the intestinal cell. And it stays there, but it does not come in the blood because already there is too much iron. It can harm the cells. So ferroportin is degenerated. However, reverse happens. If transferrin saturation is low, that means low amounts of iron is in the blood, liver gets the message, and then it upregulates the formation of hepcidin. It downregulates. It downregulates formation of hepcidin so that ferroportin stays where it is and it can absorb lots and lots of iron into the blood. Okay. And now this gets more complicated. What is happening is hepcidin actually has got vitamin D receptor on its structure. So we now know that vitamin D suppresses hepcidin expression. It might be happening at the gene transcription level because we learned how important vitamin D is for DNA and gene expression. We still don't know. These are new data. So if somebody has inflammation, that means they have some infection, and inflammation means a lot of cytokines, interleukins, they are moving around in the blood. Then they also can have anemia because in that status, hepcidin is down-regulated. Okay. Questions, comments? So do we know what does hepcidin do now? When is it produced by the body?
And too much transferrin, Nancy, too much iron in the blood.
Too much iron. Thank you, Kathleen. And what does it do? What does hepcidin do?
it down or stops the transferrin, doesn't it? The transportin, the
Ferroportin.
lateral layer that takes in.
Okay. Yes, yes. So it degrades ferroportin so that no iron comes inside the body or blood. And the tricky thing is there are other ways as well in which vitamin D can modulate iron metabolism. One is simply by controlling hepcidin, because it is sitting there and controlling the gene expression of hepcidin at the liver.
Nancy, is it correct? I think we can't, other than if we excreted it, you know, the enterocyte sloughs off with iron, there's no other way that we can get rid of it. Is that correct? If it's, you know, if we absorb too much, is that correct, Nancy? There's no other way we can sort of, and that hepcidin is in place, because once the iron is in the body,
yes,
excrete excess levels, can we?
Yes, that's a good point, Kathleen. Yes, that's why body has evolved to take control of that iron saturation in the body. So what happened if somebody, you know, like in the case study for question 2, somebody who's obese, they have a lot of inflammation happening. So that actually upregulates vitamin D because it's fight that inflammation, and that kind of makes too much hepcidin. Okay, and because hepcidins, why again? Because hepcidin has to prevent iron to come into the system, because too much iron will add to the inflammation. It will further cause the damage to the cells. So from that level, vitamin D is crucial for iron metabolism also. And we also have now data that shows people who are deficient in vitamin D are also deficient in iron. Whereas this was observed only for, vitamin A and iron earlier. Yes, Amanda, you are right. When there is too much iron, inflammation triggers the hepcidin too. Yes, so there are 2 points. And if you will go through the research paper attached to that assessment, you will find that vitamin D influences iron metabolism even at the erythropoietin level. So that's what the activity is about, that you have to find out other mechanisms through which vitamin D can influence iron absorption. But please ensure to discuss hepcidin really well there. Okay. So the slides, it's listing some of the dietary inhibitors and enhancers, and you are all aware about it. So that's okay. I'm taking other questions as well if there are regarding iron absorption. So Tracy is asking, I believe that that's where one get anemia of chronic disease, which is hepcidin related, not from lack of dietary intake. Yes, Tracy, perfect. Yes. And yeah. And Talia is saying, to go over the role of excess inflammation in the process again. So what's happening is inflammation, when it happens, there are some inflammatory mediators called as cytokines. You might have read in some of the research papers about tumor necrosis factor alpha, interleukins, and we'll go through that in session on inflammation. So when that is happening, in the process of preserving our cells, we can also harm our own cells. So there is a lot of fire in the body in a way, and it is harming the normal cells also. Now, this is a condition in which iron can become really naughty. It can also become an oxidant. That's why in this process, hepcidin manufactured by the liver is enhanced. And hepcidin then goes on to fight off ferroportin so that excess iron does not enter the body. And till I read about this, I always used to find women mainly who are obese, they have iron deficiency also. And I couldn't understand till then why it is so. And it makes sense now that it is all controlled via hepcidin. Does that help, Talia? Okay, so let's do manganese and then we'll take a break. Okay, 5 minutes break. So manganese, it is found in legumes, coffee, tea, and I have my notes that are saying that even wine has got little bit of manganese. Not that I am telling this to my clients to have wine for manganese. So its role as other family of ions, minerals, it also is a constitute of metalloenzyme. And there are various enzymes in our body ending with lysases, ligases, kinases. They all can be activated by manganese. But mainly those enzymes, they need iron or zinc. But they can use manganese also if the others are not present. But that doesn't mean that deficiency of manganese can inhibit these enzymes. So our body has ways to use iron and copper instead. So now manganese absorption occurs as Mn2 plus. We don't know why, but it has been noticed that females, they absorb more manganese than males. A mystery. Though I know we may all try to understand it through something, but yeah, it's tricky. Then at brush border, same transporter, divalent mineral transporter 1 absorbs it. Then, though we are still understanding, but they are saying even the transporter for zinc, which is ZRT-like protein, this is mainly zinc transporter. That can also takes manganese inside the enterocyte at brush border. But at high doses, simply diffusion can also enable manganese to come inside the enterocytes. At the basolateral layer, we don't know how manganese is coming into the blood, but mostly we are carrying it with albumin or macroalbumin, and sometimes it is also found attached to ceruloplasmin, which is a copper transporter, and transferrin, which is an iron transporter. So there is a chance that if I am taking manganese supplement in higher doses, I am allowing for competitive inhibition for iron and copper absorption. Now, again, all those complex interactions that if we look at the inhibitors for manganese, it is kind of interacting with iron and copper. Iron in excess inhibits manganese absorption, and it's the reverse also. Manganese in excess also inhibits iron absorption. Okay. So There are some deficiency signs of manganese, such as growth can be impacted, reproductive function can be an issue, skeletal abnormalities can be observed, even issues with glucose metabolism, because manganese is a cofactor for one of the uh, carbohydrate metabolism enzymes. So again, the point that we need to remember is think in optimal dosage, separate supplemental dosage. If the food forms are not working because somebody has malabsorption syndrome, and yeah, just staying away from the upper limits for these microminerals or trace minerals. Okay, time for a 10 minutes break, right? Let's listen to some music and come back in 10 minutes, but I'm here if you have any questions. Okay, so I'm not sure whether everybody has come back. I'm just showing rich sources of iron for vegetarians. So this one is black chickpea. And then there are other dark color lentils. So one of you were talking about red lentils. So if you look at the figure on the right, that are black lentils. And if you remove the husk, that actually makes red lentils. Okay? You can even see that there is a glimmer of that redness. So this is the one. And if I remove this husk, it will become red. So if you're removing that outer dark coating, that means you are removing all the minerals and other good stuff, fiber. So if you're taking it for fiber and iron, go for dark lentils.
Mansi, what do you do? Oh, this might be too much to ask of you, you know, with the lentils. But for me, I have very poor digestion. I'd love to eat more lentils, but I know I find them difficult to digest. And I know they say if you introduce slowly, but as regards to soaking them or cooking them in a way that makes them more digestible, do you have any tips?
Number one, go for only husk-free lentils. So red and yellow lentils are the ones for you. Okay? Don't go after the heavy ones, such as even chickpeas or these black ones. You know, if somebody, I trained as a dietitian in India. So if somebody has a liver condition, if somebody has a digestive condition, and most of the people, they don't take meat. So we give them red lentils that are soaked and just boiled and added with some olive oil or medium chain fatty acid. So it is easily absorbable. So for you, it would be simply having red and yellow lentils. Okay. And regarding soaking, always soak the red and the yellow lentils for about 2 hours and the dark lentils overnight for 14 hours or so, and then throw away that water. So that's where we are removing all the factors that may cause indigestion and bloating.
Thank you, Matt. I've just got some mung beans and I've been trying sprouting them as well because I've heard that that's
yes, I have them in the kitchen right now. So when you are sprouting it, you are increasing the iron content, you are increasing the protein digestibility, and you are increasing vitamin C for sure.
Okay, no, thank you so much, Nancy. That's very helpful.
Yeah. But for people who have digestive issue, it's better to steam a little of those sprouts. I know it's killing some of the benefits as well, but it's better to avoid that digestive issue.
Yes, no, thank you.
Thank you for just riding with me on this ride to understand our body and to understand our connection with food, isn't it? Okay, is everybody ready for the next session? Let's move on. So now we discuss about zinc. Zinc also can exist in several forms, but the only one that we need to remember is zinc 2+. It has various functions again. I have talked a lot about superoxide dismutase and metallothionein, which are our body's fighters of infection. It is required for growth because it participates in the transcription. So, you know, all the enzymes, when we are expressing a gene, we need some enzymes, and all those enzymes need zinc as a cofactor. Another important point, we keep saying that we are exposing our DNA to pollutants, to heavy metal. And you know, because we are a very complex being, there are so many cells, body is always dividing those cells. And when we are making our DNA, there can be some mistakes. So when there are those mistakes, our body has something called as DNA damage sensing enzymes, and then we also have DNA repair enzyme. And all those enzymes need zinc as a cofactor. Okay, so that's the thing to remember from clinic perspectives. Zinc is also required for heme synthesis. And remember we talked about phosphatases, B6 enzyme and niacin. When they have to be released from food, we have to separate phosphorus. There also we need zinc. So I'm speaking other benefits than what are listed there on the slide. So you can imagine how important zinc is. When we are fighting infection, we need zinc because it is, okay, what I found was that when we have an infection, body makes T cells, and then body decides, you know, okay, this is a virus infection, so I have to make macrophages. No, this is a bacterial infection. I have to make cytokines. So all those T cells actually can take the shape of fighters with different capabilities. Some have swords in their hand or some have other stuff. So again, I'm trying to simplify. And that happens only when zinc goes and binds to a particular enzyme that does that. Okay. So zinc, that's why zinc is so important for immunity. Amazing, isn't it? So I've just added this screenshot from a research paper which talks about how zinc is supporting. I was talking about DNA damage sensing mechanism. That's what I am hoping you will study in your other subjects about P53. So again, prevention of cancer and managing of clients who may have had to deal with breast cancer, think about zinc deficiency and how we can support those body's mechanism to sense DNA damage. Okay. So this was all extra bit, guys. Coming to zinc digestion and absorption. So the first thing is now easy. I'm not going to repeat that again. At brush border, we have got ZRT and IRT-like protein. We call them ZIP4. So remember that for your exam. that zinc is absorbed at brush border by specialized transporters called as ZRT and IRT. But divalent mineral transporter can also absorb it. That's why zinc may compete with iron, manganese, and other nutrients. But at high levels, if I'm taking my zinc supplement because during the flu season I am susceptible to infection, then I can take a particular high dose and it will it will get absorbed by paracellular diffusion. Again, when zinc comes inside the cell, our body has the capacity to use it. It can be stored with copper as a metallothionein, or it can be used to fight any infection, functional uses inside the cell, or it can be stored for some time in the Golgi network. At the basolateral layer, we have zinc transporter, and in the blood, zinc moves around attached to various albumin. So that's kind of very similar to most of these divalent ions. Okay. Now, factors affecting zinc absorption. Some of these you might be familiar. So vitamin C, because it helps the transporters. Amino acid, because it's bind to zinc to store it. Picolinic acid. Have we heard about this one? Okay, you might see that there are some supplemental forms. They are present with that. Chromium picolinate, zinc picolinate. So our body makes it in the tryptophan metabolism. Okay, and this picolinic acid, it is a good chelator. I have used this term again. Chelator means it binds it, it protects it, and does not release it till it reaches the point of action. So that's why, zinc picolinate is the best form of zinc supplement for a client, and even chromium picolinate. Okay. So we have realized that our body makes it, and now we are using it to make some good supplements. Low zinc status is an enhancer because body knows it and body tries to absorb more of it. So if I am zinc deficient, body will absorb all the zinc supplement that I am taking. Antacid, PPIs, again, for all those minerals, they are inhibitors because they change the acidic milieu of our gut. Divalent cations, again, it must be clear fiber because it binds to zinc and excrete it. Why? Okay, folate is listed there. Why is it an inhibitor? So my understanding is that, remember, when folate is getting converted to monoglutamate form at brush border, we need conjugases, and those conjugases are zinc dependent. Okay. So there is a possibility if you are consuming too much of polyglutamate, then body may utilize zinc to convert all those forms to monoglutamate. But it should not be an inhibitor. It should be kind of there should be a separate category that it may increase the demand of zinc, isn't it? So yeah, it should work in that sense. Any comments, any question at this stage?
Nancy, might I just ask you a question? You know, with these trace minerals, how they go into the blood and they're always attached to a protein. Other than free iron, it's dangerous if it's traveling free. It can be a pro-oxidant. But
Yes.
why is this a silly question to ask? Why they can't travel on their own in the blood? I understand the fat-soluble vitamins because they're not dissolvable in the blood or and transported. But is it a silly question to ask why, other than iron, why these others need to attach to a protein to travel around in the blood?
Kathleen, there is no silly questions, but there can be silly answers.
Yeah.
And like the one that I'm going to give you, that I really don't know.
No, it's just the way it is.
Yes, and like again, my understanding is body treats them as precious.
Precious.
Body
Yeah.
knows if they move away, they may interact with other oxidative products moving around in the blood or in the body. You know, we may have a lot of free radicals. We may have a lot of inflammation in the body, and as soon protein bond is lost, iron and zinc, they may become oxidative. So again, that's what my understanding. So body is trying to stick to them till they deliver it to where they are needed, which may be in the immune cells, which may be, you know, in the liver when we are detoxifying. So yeah, I can, so that's the silly answer that I have, because I can't attach a logic to it completely. Sorry.
No, that's fine, Nancy. It makes sense what you're telling me. Thank you very much.
Welcome, Kathleen. Does anybody has any confusion at this stage? Do you want me to repeat anything? Okay. So moving on, we are now talking about ultra trace minerals, that is chromium. So you can imagine it is required in really, really tiny amount in micrograms in our body. There is a story regarding how it was discovered. So some experiments were done, you know, in the yeast, and the researchers thought that that yeast is helping the glucose metabolism. So that's how they started, and then they found that brewer's yeast had a lot of chromium in it. So its discovery started from there. They realized that it may have some role in supporting glucose metabolism and preventing insulin resistance. But now we know that some spices such as cinnamon and cloves, they have chromium apart from turkey or other organ meats. So now we know about other sources. So now coming to this slide, you can see that chromium can also exist at various states. You can see chromium -2 to chromium 6+. And it can be toxic when it is 6+. It is carcinogenic. It can be a skin irritant. So we are not concerned about this one, but our body favors the more stable form, which is the trivalent chromium, that is chromium 3+. We are not sure about its absorption at brush border. Only thing we know is that we absorb chromium 3+. There may be passive diffusion or carrier mediated transporter. We don't know. We speculate that chromium is traveling with transferrin in the blood. If transferrin is occupied, then albumin attaches to chromium and carries it to the liver and other organs that may need it. So enhancers, amino acids again, because they bind to chromium, make it available for its action. Lipophilic compounds such as lipoproteins, they may also help. We have noticed in some trials that vitamin C intake increases chromium absorption. Okay. Now, just one more clinically relevant thing which I wanted to discuss in respect to chromium. Sometimes some people have to fight their hunger and their sugar craving. That's why think adding cinnamon powder on their breakfast, cereal, or any other, even salad, it may not feel good, but with any cereal, it will go really well. It reduces sugar cravings. Okay, so think chromium picolinate. If somebody is coming to you with insulin resistance, they are obese, think chromium. We now know that in PCOS, insulin signaling is affected. Insulin resistance is one of the etiology factor, one of the causes of PCOS. Okay? And why? These are the benefits of chromium that we know through some randomized control trial. So please remember a couple of them from exam perspective. You would notice I've added this slide here just to reiterate how chromium supports insulin function. Number one, we now know that it can mimic insulin. Okay, it can potentiate action of insulin when it acts as a chromodulin. So what happened? What is a chromodulin? It has 4 amino acids, and all the amino acids can attach a chromium ion to itself. So that forms the active form of chromium. That is chromodulin. And this chromodulin can potentiate, that means it can increase the function of insulin receptor on the cell membrane. Then chromium can also support GLUT4 translocation. Remember, GLUT4 in the skeletal muscles is inside the cell. It has to come out. So there, by supporting this signaling pathway, MAP, chromium can directly tell GLUT4, come on, go outside, there is glucose to come to absorb. Okay, so there are benefits, and you must be thinking now how much dosage to give, in what amounts. So some of the trials that are listed here, they used a dosage of 200 micrograms of chromium picolinate. Some of them even used 1000 microgram per day. But again, for my clinic, I would not go up to 1000 microgram because I'll be looking at some other techniques, other nutrient intervention to support insulin resistance, isn't it? We'll be telling them to exercise. We'll be telling them to take low GI foods. So along with that, chromium would be my supportive strategy. So I'll stay with only 100 micrograms or so. Okay, comments and questions. They are doing okay. A couple of nutrients left. This one was easy, less transporters to remember, isn't it? Yeah, tell me.
Nancy, I was just going to say, so, cinnamon has been renowned, hasn't it, for helping with insulin resistance, and that is because of its chromium component, is it?
That's our understanding so far. But, a couple of years later, we may found that there are other goodness there, like for turmeric. Earlier, we were aware that it has only curcumin, but now we know turmeric acts best in its whole form because there are other antioxidants. It's not only curcumin. So same way, I expect that if we do more lab analysis, we'll find cinnamon has more good stuff. So, but yes, right now our understanding is that cinnamon has chromium and it helps and addresses sugar cravings. But in tiny amount, isn't it? It can become really bitter if you use it in a high amount. Like if you go up to 1 teaspoon also, it can be really bad. So stay with, couple of pinches of cinnamon. One thing that always I share with my students is that when we are talking about herbs and spices, try to think how their absorption would be enhanced. So think about a good fat source. So think about sprinkling a little bit of olive oil as well, because these herbs, they have a structure like turmeric. Again, I'm depending what I'm saying on what I know about turmeric, that it is best absorbed when it is with fat, and it has to do with their chemical structure. Okay, the question that Kathleen you asked the other day about ester molecule. So yeah, it's something to do with their structure, that they are absorbed well with some fat.
And Nancy, turmeric as well increases with the pepper, doesn't it? Black pepper.
Yes, pepper is another one that supports it. Yes. Again, a story. Sorry if I'm boring you. So, even today, like in my family, if a child is born, if somebody had a delivery, then my mother-in-law will prepare something, like she will give me ghee and she will add lot of turmeric in it and some nuts. And it was many years ago. It was actually 20 years ago, and I was fresh from my dietetic degree, thinking all those bad things about the ghee. And I was crying and I was fighting with her that I'm not going to have it. So, and now, when I'm teaching, when I'm researching, I find out that there is a logic behind that, to have that turmeric during that time, because turmeric is an antioxidant, it is an antiseptic. And always, if somebody has arthritis pain, my family, they will just have turmeric in milk. I know milk can have an issue, but for arthritic pain, they will have turmeric in milk, and they say it works. But if you look at the randomized control trial, you might not find such positive result. So that's where we try to make a balance of anecdotal evidence and a person's individual health situation, and then what the clinical studies are saying. Okay, sorry about that story. We are really digressing a lot today. Let's move on to iodine now. And we'll discuss the questions in the chat after this. Iodine, again, many forms. So we have iodide, and then we have iodate. And this is the form that is added in the iodized salt. So we have potassium or sodium iodate as part of mandatory food fortification in Australia. Functions, we know that it is required for synthesis of thyroid hormones, because it's the two iodine molecules that have to attach to tyrosine to make thyroid hormones. And there we also need vitamin A, and we also need iron, because they support those enzymes that are making them. Again, for absorption of iodine, we are not sure how it is happening. We know that it is only the free form that must be absorbed. Supplemental forms also, it's kind of very easily absorbed by simple diffusion. Okay, that's what we are aware, but I suspect in coming years, we may find that there are other transporters. At the basolateral layer, again, we are not sure. If it is a supplemental form, it will come out simply by diffusion. But the point to remember from exam perspective and otherwise is that when iodine reaches the thyroid gland, that's the place where it is used maximally. And there we have an active transport mechanism happening, and we call it as sodium iodine symporter. So for one iodine molecule, a sodium is exchanged. So that's why for enhancers, we have tyrosine and protein and glutathione, because glutathione is a major antioxidant even in the thyroid gland. So it kind of supports this mechanism. And deficiency of these micronutrients can impact thyroid function, and it goes together with iodine. That's why it is written as inhibitors. Whereas again, as I'm saying that I should correct myself, they should not be inhibitors. It's like their deficiency may cause an issue with making thyroid hormone in our body. Selenium also supports this pathway because it is a cofactor for glutathione, and we'll discuss about it in subsequent sessions. And I'm assuming we are aware about goitrogens. Your turn.
Nancy, I know they're in some I think has broccoli got some in them, and yeah, they impact.
Yes, perfect, Kathleen. So these are the cruciferous wedges, and they inhibit iodine uptake because they have these compounds called as isothiocyanates. So tell me, if I have a client who is coming to me who is hypothyroid, but she wants to go on liver detox. And in liver detox, we love broccoli and cruciferous veggies. What would be my priority? What will I tell the client? Okay, so Jane is saying cook them. Yes, so just steaming them, not enough, because again, that will kill the good antioxidant, because these isothiocyanate, they actually even affect estrogen metabolism. So clients who are trying to be proactive in preventing breast cancer, there also we want to educate them to take a lot of cruciferous. So we can use other antioxidants, we can use other compounds to support thyroid. We can steam it. And yes, we can sometimes prioritize hypothyroidism. Correct that, and then go on to proactively work with prevention of or estrogen metabolism. Okay. So there was some interesting discussion again in the chat before I started iodine. Oh. Okay. Thanks for pointing that mistake about T4 and T3 in the slide. I'll have a correction at it. Thank you, Jane. Tracy was talking about micromineral depletion in the soil. And she's asking whether we need a supplementation will increase and food fortification. What choice should we make? Or we simply go for organic? Yeah, definitely. You know what I've been telling my husband, that he's growing some, he's very good at all these stuff. I'm not very gardening fan. He's growing some herbs, coriander and chilies and even beetroot and all. And I'm telling him to get some soil which is rich in iodine. So get some seaweeds. So just trying to keep that balance, give the soil its balance that was missing. Okay. So did that make sense, Tracy? So you have a point there about thinking about our soil and
Yeah,
way.
yeah, I think it's just interesting because it's not something that we consider when we're thinking we're doing the right thing by buying vegetables and fruits and things from the supermarket. You don't really know what content of these micro minerals are in there.
Yeah.
And I guess it's just the big farming methods that are used and things like that these days. So yeah, I think it's just something that I've been thinking about, and whether buying local from smaller farmers or yes, growing your own, obviously that would help, and you've got a bit more control over the soil content.
Yeah, change is happening slowly but surely, and I always have great hope from all my students. They are going to be at various places in public health, as nutritionists, even in our homes. We are making a change when we are doing it for our family. We are discussing and educating our family members. We are doing it with our children. So every step counts.
Yeah, I agree. Yeah.
Okay, now Jane also has another important point. Iodine and high thyroid antibodies is an issue. Yes, yes, that's an issue. Thyroid is very sensitive, and again, a story, guys. Sorry. So, about 5 years ago, Adelaide summers, and I'm watering my plants, and I had a tick bite. Okay. And it led to some symptoms which were similar to rheumatoid arthritis, but nobody could diagnose it. It stayed for many months, actually. I had cruciating pains in my joints. Okay. Then following, now I can say it in that sequence because now I know. After a few months, I was diagnosed with hypothyroidism. And nobody was interested in knowing why I had it. It was actually quite recent, that I got those antibodies done and realized that I have them. And now I'm thinking that it may be that it's because of that tick, the inflammatory response my body had, that it started making thyroid antibodies as well. That may have led to hypothyroidism. So those of you who are not aware, just relax. You will study about that in your clinical subject, that one of the reasons of hypothyroidism could be that our body starts making these thyroid antibodies because of some other factor in our environment. Okay, so relax. Don't. Sorry about the story, but again, intention was that it will connect when you study about clinical subject. And thanks, Jane. That was a very good point. Okay.
Nancy, what can you do then about these high thyroid antibodies and impact or causing you to have hypothyroidism? Do you mind if I just ask you that?
Selenium, guys. So selenium helps because selenium supports glutathione. It itself is kind of an antioxidant.
Okay.
And glutathione is our body's endogenous antioxidant. It is able to keep a tab on and that inflammatory reaction. So 2 drops of selenium drops to such people for about 3 months, apart from supporting thyroid activity together.
Yeah, and you're able to, your thyroid's okay then now, Nancy?
It's better. It's better. I've been taking selenium drops for some time now.
Very good. Very good. And Nancy, also with you were saying So if you cook those cruciferous vegetables,
Yeah.
the iron, that stops the iodine inhibition. Is that correct? By cooking the cruciferous vegetables, those that destroy the ice. Lovely.
Yes.
Thank you. Thank you.
Yes. Where is that term written? Isothiocyanate, that was the one. No? Yeah. Oh dear, I'm sorry about this. Yes, that's what it will do. Okay, moving on to molybdenum. Again, various forms, MO4 plus and MO6 plus, that's their chemical names. Mostly it is bound either to a sulfur or an oxygen. We don't know much about how it is absorbed. So we are just assuming that there may be some carrier-mediated mechanism. In the blood, we are assuming that it must be bound to albumin. So the point is that we don't know much. Selenium is that important trace mineral about which we'll talk a little more. Again, it has some organic forms and it has some inorganic forms. So we have selenomethionine, selenocysteine. Now you must be thinking that this looks like some amino acid. So you are right. Selenium actually exists in nature and in our body in a form called as selenoprotein. So different amino acid can bound to selene. Specifically, you remember methionine or cysteine are sulfur containing amino acid. So selenium, how amazingly, can replace sulfur in these structures. And that's how it exists in our body and even does its function in our body. So different enhancers, vitamin A, C, E, and reduced glutathione. Again, enhancers because it is utilized to help these antioxidant to function. However, heavy metals, they bind to selenium. So they are considered as its inhibitors. So for absorption, we know that organic selenium can simply get inside the brush border by amino acid transporter. But if we are eating some inorganic selenium, which is actually quite rare, then we will first convert this selenium into selenite, and then that will be absorbed either by a sodium dependent transport or by passive diffusion. Its transport at the basolateral, again, we are still discovering, but mainly in the blood, it will move along with some amino acid, specifically selenomethionine or selenocysteine. In fact, your glutathione is actually a selenoprotein because it uses selenium as a structural component. So that's fascinating. All these sulfur-containing proteins in our body can use selenium and thereby prevent or manage any kind of heavy metal impact or liver detoxification, because in liver detoxification, glutathione is again our body's free radical quenchers. So we need little bit of selenium there also. And then we have the last one for today, boron, just a single slide. That doesn't mean that it is not important for humans. It's only because research has not yet identified a clear biological function of boron. However, there are some beneficial impacts on reproduction and development, calcium metabolism, need boron. There is some evidence now it is required for insulin function also, immunity. It supports vitamin D and estrogen in some way. So very little is known, but it's already in the supplements. So, okay, regarding absorption, again, we are not sure. We are assuming that there might be passive diffusion. And there are different forms moving around in the blood. Again, not sure that these are the only ones. So that's why we only have just one slide here. Okay, we have time for questions, comments, and any other discussion if you want to have.
Nancy, I think you're very clever with your thyroid because, you know, a lot of the time with hypothyroidism, people are put onto thyroxine, aren't they? The artificial
Oh, that does.
done to you
That
to
does
get
slow some time, you know.
Yes.
If we are not storing those tablets at the right temperature, it may become ineffective. Nobody
Yeah.
told me that.
Yeah. Yeah.
And it never helped me. What helped me when I started adding those, you know, dried wakame leaves, they are available. They are a source of iodine.
Iodine. Yes. Yes.
So I started adding those in my salad. I started taking selenium drops. And I shift my dependence on animal meat. So again, I'm on that philosophical path. I sometimes become non-vegetarian, vegetarian. When I increased my protein, that helped. These are the things that we clinical nutritionists and naturopaths can discuss with our clients. Yeah, because people forget about these small but important points. And I also recommended selenium to a person who had that skin allergy, eczema kind of issues. I suggested selenium to them also, just because it supports glutathione. Why not strength? So if somebody is susceptible to these allergies, that's telling me that their own immune system needs some help. So I'm very open to work with selenium with these such clients. Okay, any other question regarding selenium? Because I may have been a little fast in talking about that. Okay, yes, Jane, that was in my mind that I will post it on Loop regarding Easter Monday lecture. So we won't have any lecture. We'll all be spending a well-deserved holiday. We all should be relaxing and having some fun on Easter Monday. So I'll be recording Thursday lecture on that day, Thursday morning one. First one, it's not morning for us. Okay, so recorded lecture would be made available on, I think, Thursday late afternoon. They are pretty fast or hectic. They are doing it the same day, I have noticed. You can attend any other lecture. Tuesday we have a lecture where Larissa takes it. I'll be doing Thursday morning as well as evening. So feel free to join us. I don't want to miss you, my Monday class.
Nancy, just also, you know, you chatted about these selenium drops. I know you said you just take the 2, but because we're not sure of its, well, the absorption pathways aren't overly clear, Nancy, there's no sort of risk in it. There's no sort of competitive inhibition. Is that right with the selenium? I mean, we do know about the transporters, but yeah, it doesn't seem to compete with anything else.
Yes, there are a couple of questions here. Jane is also asking, is too much of selenium an issue? And the point that you are raising is that might be a competitive inhibition because we don't know yet. Yes, excess could be an issue, but selenium drops comes in organic form, so it will be simply absorbed. So we need to be mindful to carefully monitor the symptoms of our clients and don't ask them to take it more than, 6 weeks. Take it for 6 weeks. How is the absorption? Then continue for another 6 weeks and then stop it. Long back, I read a research article where they measured the amounts of selenium in the blood and the serum. And what they found was that body knows. It absorbs it in its micromolecular form. And if it is in excess, it excretes it. However, if we take the organic form in higher amount, selenium can be toxic. Okay, and if there is an exposure to some metal ions, then we will need more selenium. But that doesn't mean that we should not monitor it. So don't go beyond 3 months. That's my approach. Okay? So either ask them to take it only 3 times a day, and second, stop it after 3 months, observe symptoms, do your blood analysis, look at thyroid function, wherever you want to give support, look at liver detox. So please remember, too much selenium can be an issue. Tracy is asking, I believe just 2 to 3 Brazil nuts per day meets RDI. Yes, they are quite big. I think so, Tracy, you're right. But, in our class, we are always questioning and we are always asking, do we know that those Brazil nuts have the same amount of selenium that they used to have earlier? How do we know that? Some researchers need to test it. But
Also, the Brazil nut, they'd have the phytates as well, wouldn't they, Nancy? So do we need to soak them first?
Good point. Thanks, Kathleen. That's such a relevant point. Yeah. Soaking nuts always help, yes. That's what they say, isn't it? Activated almond, and now we have even milk. Activated almond milk or pistachio milk is also available now. Okay, I hope we are not digressing. We are okay. Everyone is okay with today's lecture. Again, I agree, a lot of important points, but I hope we all went slowly and we made some important connections. We made our notes and go through the slides again. Thanks, Jane, for pointing that mistake. I'll correct the slides. Kathleen also pointed something in her email about a mistake. I will change it for the next semester so that people get confused if I change something major. So, well, thank you for today's sections. Have a good week, guys. Bye for now.
Thanks so much, Nancy. See you next week.